Done, sir.
Asim sir, please proceed.
Good evening, everyone. Today we gather here for the NRI lecture series. We welcome Dr. Arvind Deshmukh sir, President Microbiology Society India. We also welcome our NRI lecture series chairperson, Dr. Chandratre ma'am, and we also welcome our resource person, Dr. Moumita Sarkar ma'am, for his valuable guidance. So first of all, we introduce our resource person briefly. She done her BSc in biochemistry from University of Calcutta. She also done her post graduation in the same subject, and she also done his PhD in biotechnology, cancer biology, University of Calcutta, India, and she is the research associate, National Heart and Lung Institute, Imperial College, London, UK, presently. So I request her to give her talk and enlighten the gathering. Over to you, ma'am.
Thank you very much. And I really appreciate and thank you everyone for giving me the opportunity to discuss my work and my journey and inspire everyone and also share my knowledge. First of all, I would like to thank all my respected seniors here and Dr. Arvind and all of you here for giving me the opportunity. Thank you very much. I hope, I hope it will be nice and fun and interactive. And please feel free for any feedback, questions. I hope you will enjoy. So today my topic is discussing about the state-of-art techniques in molecular biology and the current advancements all over the world and whichever technologies are really, which is very like in the market and which people are really thriving on. So I will start briefly with, so this will be a talk about some philosophies, and some scientific work I have done, some discussion about the protocols and the techniques, which is general, not for any specific scientific field, but it can be for microbiology or biochemistry or molecular biology in general, applicable to any sector of diseases, whether it's cancer or it's infection or whether it's a cardiovascular. And we will also talk about briefly about some career opportunities and some philosophies, just to keep it interactive. So yes, I hope you will enjoy. So first, as so I mentioned that I will talk about my brief journey of how I moved from India to London. So initially, I did my schooling in St. Thomas's Church School in Kolkata and Howrah. And after that, my initial interest was to, when I finished my school, that time in West Bengal, in Calcutta, there was no like a course which taught microbiology. So there was I was very, very interested that in that times you have to go to Bangalore to study microbiology. So I was really pursuing my father that time to really, so we were working together so that I can move to Bangalore. But luckily, that time, it was that year when everything opened in Calcutta. So microbiology was there a bit earlier than biotechnology, biochemistry, everything that year opened in Calcutta. And to cut the story short, then I moved to Ashutosh College to finish my biochemistry, BSc in biochemistry. And then from there, I went to University of Calcutta, Balligan Science College to do my master's in biochemistry. So while I was doing my master's in biochemistry, one of my special course was cancer immunology. So to be very honest with you, when I was very inspired by the teachings, and when our sir, he used to teach us about signaling and what happens in the cell and how proteins and DNA operate together to relay different kind of information in the cell. I was deeply, deeply, deeply motivated by that. And till that, I never knew I would be doing a PhD. I was like going with the flow. Okay, let's see what happens, what I like. And then after that, we were a class of 60 students. So in Masters of Biochemistry, and I think 50 are in the States now. So as we going with the flow, we all appeared for GRE. So I appeared for GRE. I was about to go to America to do my PhD, and I got an offer from that time Chicago Medical School with a scholarship of $20,000 that time. But then that time there was a bit of recession, if I remember, and my mother was not very happy. She was like, "Oh, you're going for the first time. There's recession. I'm not sure you should go." And then I rejected my offer because my mother was not happy at all. She was hell bent that I don't go. So after that, I'm like, oh God, then. So you spend 6 months preparing for GRE, giving the examination, getting an offer, and then saying bye bye to that. And nevertheless, then I was lucky enough to clear net by a go because I had all the time. So, all the time, 6 months I just studied. And one of some seniors also, like that time I was doing an internship in IICB, Indian Institute of Chemical Biology in Kolkata. So there I was working on Vibrio cholera, and we were working on an environmental strain versus the pathogenic strain, and what is the difference between the sequence in the toxin gene and why that is environmental and why the other one is pathogenic. So that time, some of my seniors, so I was working with Dr. Rupak Bhadro that time, and some of my seniors really gave me very good advice, like how to crack net. And they said, okay, you're not going to US, fine, come on, there are a lot of good opportunities in India. So if you want to crack net, what you do is answer as much as possible, whatever you know. Doesn't have to be perfect. Try to be scared. That time there was a written part as well. As much knowledge you have, just pour in there and try to make diagrams and schemes so you just crack net. So it just worked out for me. I think I was lucky and I just got the net for the first time. And then my journey started, my academic journey started. So because I was based in Kolkata that time, I looked around and I saw Dr. Mrinal Ghosh. That time he was quite new. That time he just returned back from the States after a long scientific journey, and he used to work in Cleveland Clinic. And after that, he came to IICB and joined as a scientist. Like, he was quite new that time. I think he was probably three years in IICB. And I looked at his work. It was more about cancer cell signaling. And that's what I always wanted to do. And I was like, okay, then I went in and I said, sir, I want to do a PhD with you. He said, oh, so did you clear it? Do you have a fellowship or something? Are you sure about that? I said, yeah, I'm pretty sure. He said, okay, come in, let's have an interview. So that time I think I attended 4 of his lab meetings. He was very tough. That time he used to be very strict. I mean, 4 interviews. Like every time I used to go in his lab meetings, had to listen to all the lab meetings and then have to summarize at the end. And they would ask. So finally, after 4 interviews, he finally decided to take me. But it was quite nice because everyone was asking a lot of questions. And henceforth, I started my PhD. So my
So you
topic.
qualified net or net GRF?
Net GRF, yeah, the CSIR one.
But yes, because in net LS there is no fellowship. That's why
Yeah, Net GRF.
confused.
CSIR JRF net.
Yes, CSIR GRF.
On CSIR laboratory to do PhD, you need CSIR JRF or UGC JRF net.
No, CSIR net, CSIR GRF net. Yeah,
Wonderful, wonderful. Who was your guide, you told, by the way?
Dr. Mrinal Ghosh.
Okay, please go ahead.
Yeah, he was, and that time he was really strict. And that time, he was like, come to all my lab meetings, listen to all my lab meetings. Hence, that time I met Malini ma'am as well, like there as well. And then I was like, and everyone was then, taking an interview for 4 lab meetings. And sometimes I won't be able to answer the questions. And he will be like, oh, so you don't know these answers. And everybody from the lab, like, sir, she didn't even join. Come on. And then, anyway, eventually I joined his lab. And it was very interesting. I think what I learned from my PhD was very interesting. It has been one of the most important. I spent 5 years in his lab completing my PhD in cancers. So my topic was that time to study an RNA helicase. It's called P68. And so that time we just had the clone. It was sent to him from Scotland. He said, Momita, this is another person who's working with me. This is the clone. You study, you read, and you say what you want to do. It was not optimized at all. And then eventually, at the beginning, my senior who was with me in the same project, he was too senior. He was too busy. He was almost about to finish. So he didn't have that much of patience to teach me everything. He used to get angry a bit, like, how are you doing mistakes and all. So eventually, he told sir that, oh, she's too, too junior. So then my supervisor said, doesn't matter, Momita, you do independently. You come. So I think what really helped me that time I used to think that he was too rigorous. But now when I look back, what he used to do to me at the first year was every Friday he used to call me in the evening and he used to tell me, you read a paper, come and just present in front of me. He was busy. He used to work. I will just go present the paper in front of him. And every lab meeting, we used to have journal clubs, and he used to encourage all of us to present good quality papers. Like seniors would present most like lab data, but the people who were more junior, early years of PhD, we would read good papers and try to present, and we would discuss. I think that really helped in building the scientific hypothesis, how things work, what are the good things required to do a good work, publish a good paper. That really shaped. And then also knowing about the techniques, because we did everything from scratch in the project. So like troubleshooting, things failed, things didn't work, because I was trying to work in a normal cell. And then when I started after 2 years working in cancer cells, the phenomena was completely opposite. So it was a thing. And then I cloned so many genes and promoters. And for the first 4 years, there was no papers. I was just doing a lot of things. But at the end of 5 years, I think it was very successful. I ended up publishing 8 papers in total. There was 2 first authors in Oncogene and one review, and then other. We were very collaborative. All of us used to help each other a lot. And there was healthy competition. We would help with each other. We'll help and we will discuss. And what I really appreciated that time in my sir was like, even I was very junior and other people as well. He would pass the manuscript of the paper to everyone to have a look, to give some input. I think those things really helped, because he would not say, oh, you're too junior, you don't need to look at a paper. But that really helps, because you build up your knowledge, you see how a paper look, and everybody gives some input, everybody gets some paper. So I think it was really, really good. It was a very good journey. I really look back and I think I learned a lot of resilience, independent work, independent thinking, independent scientific hypothesis generation, independent execution of project, which now is benefiting me a lot when I look back. After that, I moved. So after finishing that 5 years and a very successful PhD, I was applying again. I wanted to go to US, because the kind of work I used to like, more like cancer signaling and everything, was some of the universities I liked in America. But eventually, I got some offers. I didn't like the labs. Then I liked some labs. They didn't like me. Eventually, it was Northwestern University, which both the parties liked. Like I liked the lab and they liked me as well. And then in the end, I got an offer from Northwestern. Before, I got an offer from Yale in a telephonic interview, and the person said, you have good papers, you have done good work, I'm happy to offer you the position, but you have to work throughout the week, like 24/7. That time, which I felt I would more, and I was a very hardworking person, but because you are new to a country, and when we are in India, we have our families to take care of your things. But when you are in a new country, you have to cook, you have to take care of yourself as well. So 24/7 is literally like, because he said he's a clinician, he doesn't have time to come in the week. So he comes in the weekend. So everybody has to be there throughout, like Monday to Sunday. I don't know. I didn't like that idea and I didn't take up that offer. But eventually, I liked Northwestern University. But my destiny was something else. So after a few weeks, the professor called me and said, sorry, Momita, my grant got cancelled. It's very common in US. I think most of you know that sometimes their grants gets cancelled. It's R011 randomly. So their grant got cancelled and he said, I cannot hire you, but I'll talk to you later. In Europe, in London, I didn't apply because I didn't know any person in Europe that time. Not a single friend I had, not a single family member. Sometimes you have these job advertisements. I just applied on the side. I'm like, okay, and I got an interview from Imperial College. And eventually they interviewed me and then they said, okay, Momita, we give you the position. And I was still in doubts, I was like, not too sure. And then my father was like, why are you wasting time? You just spend 1 year just applying and not going anywhere. Now you got the offer and now you don't want to go. And he was bit like, what the hell? I said, okay, fine, And then eventually I moved to MRC. So initially, MRC is an independent MRC Institute of Clinical Sciences. Is part of Imperial College, but it is more like an autonomous institute by the government. It's very prestigious and it's hosted by Imperial College, but it is more like a government institute in the UK. So there I came and I started working on cancer bit in more like pancreatic cancer. And also the most was stem cells. That was very new to me. So it was more like an early development thing, like how cells and stem cells form lineages in early development, in early mammalian development. It was completely new for me. I was working with a very old professor who has been in Imperial College for the last 30 years. And he was very strict, but scientifically very, very good. I learned a lot from him. He was Professor Neil Dillon. And so eventually I finished my postdoc with him for 4 years. And then there was COVID. So I was a bit unlucky in that time because there was nobody, I was about to send my paper and everything was closed. No one was allowed to go to the labs, as we know how it was. But eventually we still managed to publish the paper. Then after that, I moved to now where I work. It's with Professor Costanza Emanuele. So it is more in the National Heart and Lung Institute. Because I was working with stem cells, I was very interested to work with cardiomyocytes and adult stem cells. And then I was always interested in cancer. So that was always on my side, that when I have my independent lab, I'll start working these axes on cancer as well. What I learned from here in the last 5 years, I've just finished in May. So what I learned here from Costanza Emanuele is completely different from my first postdoc. My first postdocs, we were very few people in the lab. And I was working on one single project wholeheartedly, and we were aiming to send it into a very big journal. So a lot of work. Here, when I came to this lab, it was a completely, she's one of the chair professor of British Heart Foundation, and our lab was very highly funded from British Heart Foundation. And this is a very dynamic lab. Here, I used to work like at a time with 6 projects, different areas of cardiac repair and cardiac regeneration, lot of collaboration, lot of interaction with people from Europe, people from America, a lot of students, visiting researchers. Like from the past 5 years, I've supervised 3 PhD students, 6 master students, 4 junior researchers, lot of collaborations, lot of clinical samples from like my whole project was based on heart biopsies, like human heart biopsies, and a lot of like high level state of art molecular work as well, which I really enjoyed. And I just published my paper. So we'll talk about that. And so, yeah, and now I've just finished my postdoc. And looking back now, what shall I do? I mean, I'm applying for a couple of lectureship positions, but besides that, I think I want to transition more into government sector, more into charity sector, because in this, because I have been through a lot of difficult times as well during COVID and everything. So I trained myself as a life coach as well. And it's all about confidence, resilience, and a lot of things I learned. So I have 2 careers. I'm a parallel life coach, and I'm also a scientist. So I really want to inspire people, help people in terms of science, in terms of community engagement. So, and there are a lot of opportunities in the UK to work. When I have been working, volunteering with autistic people, coaching autistic people in charities and career development. So something like that. So I'm exploring in terms of scientific attributes in high-level government sectors. And so, yeah, that's my journey. So coming to the scientific bit. So today we will be discussing about few high-level molecular biology techniques, which I have worked with over the years and which I really found interesting. And I'm sure it will be nice for you to apply these techniques in your own field of research. So this is mainly for the students to make it more fun and interactive. So the first technique which was ever performed in this generation was isolation of DNA by Friedrich Mischer. So Friedrich Mischer, it was a discovery of nuclein. So, what he did was from some wounds and bandages, he isolated the first DNA and he isolated nuclein in 1869. He was a Switzerland-based scientist where he isolated the nuclei of white blood cells found on surgical bandages, making the first crude extraction of DNA. And that was the triggering pioneering work in molecular biology. You can see those times, the labs and the dedication the scientists had that time. It's really inspiring. And so I think before I start all the techniques, what I've learned all these years, that, what I realize is our country, all the students from every state are really, really, really good. We do have amazing brains because I think we are raised by our parents, by our society in a very good culture. We respect elders. It's rooted in our civilization. And we think, so we are known to be the biggest think tanks of the whole world, and we are very, very efficient. But sometimes what happens is that sometimes we are too efficient and we are too naive, but the world always outside India is not very kind. So they can try to, just use you for your skills and not give you enough opportunity to thrive and grow. So that is why it is very important to learn the techniques, learn experimental techniques, learn the nitty-gritty of it. But the most important thing is, it's not the techniques, it's the question. Because if you don't question yourself, why I am doing this, what is my hypothesis, you might master the technique, but you will not master the science, and then you will be taken advantage of just for using you to just do the technique, but you will not be given the opportunity to do the science and to grow in your career. Because people everywhere, it's opportunistic. You will not have your teachers, your professors, your certs there to protect you. Here, when you actually go out and fledge out, people are like, a lot of people are predators. They will just predate you of what you know. They'll use you. So it's very, very important. That's what I've learned all my life. It's very, very important that you stand on your ground. You know what you're doing. You know the science, so that nobody can take advantage of you just because you know the technique. So that you can claim that I know the science behind it. I know the question and I know the answer and I know what I'm doing. And I think that is the most important message I always tell my students, and I tell everyone, that why are you doing it? The why is very important. Everyone can do it, but the important thing is why. The question and the answer and the hypothesis. So, as Dalai Lama once said, and our monks and our generation of philosophers said that it is our responsibility as a scientist that science serves the interest of the humanity in the best possible way. So after you're working so long as a postdoc, eventually I'm sure you all, our senior professors and scientists and sirs know, but it's very important that it serves a purpose. You're just not doing it for the sake of doing it. It has to be like, the science, you do the techniques, but it has to be meaningful. So you have to drive your knowledge and motivation in that direction, that it directly serves humanity. It's just the not sake for, okay, I do this, I get a PhD, I get a postdoc, I have titles, but my work doesn't serve anything. So make sure you always have that in mind. So coming to more the scientific part. So it's just a very brief snapshot because we will be zooming in different areas of gene regulation. So first of all, and I will just be discussing few interesting techniques which I have learned along this time. So as you know, that gene regulation is very, very important. Without gene regulation, nothing works. So we have gene regulation at various levels in the cell. So first, inside the nucleus, you have the DNA replication. The DNA is produced. And here, during the DNA, you have the epigenetic regulation. So what happens is, there is chromatins, there is DNA, there is promoters of DNA in the genes. So there is one type of regulation, and that regulation is called transcriptional regulation or epigenetic regulation. So when the transcription is affected, the gene production is affected. And another is the epigenetics of the DNA, where you have some factors like, say, a methyl group might bind the DNA, an acetyl group might bind the DNA, and then it can influence the transcription of the DNA. And hence, it can influence the gene expression. And that is the epigenetic regulation. Next comes epigenetic regulation of the DNA. Then comes after the mRNA is produced. So then you have, it's called the post-transcriptional regulation. So when the mRNA is produced, a lot of regulation, mRNA can be decayed, mRNA can be degraded by several enzymes. Then you have a very important branch of science, which is very, very emerging. It is called regulatory non-coding RNAs, which will be my topic. Talk will be mostly based on that. So non-coding RNAs are, say, microRNAs, which were discovered in the late 90s. The first microRNA which was discovered was LET7. So these are the RNAs which doesn't produce any proteins or genes. They are non-coding. But what they do is they bind the mRNAs and regulate their expression. Either they degrade the mRNA or they regulate. So basically, they silence the gene expression of the mRNAs. So that is there. And apart, microRNAs were initially discovered as the non-coding RNAs initially, but now you have different species of non-coding RNAs like circular RNAs, long non-coding RNAs, enhancer RNAs, small RNAs, piRNAs. There are a lot. It's a very huge emerging field which regulate the mRNA transcription in a eukaryotic or a prokaryotic cell. Now, in a prokaryotic cell, it is a bit different. And now after the post-transcriptional processing, you have the translation. So you have the protein, and now it's most like a post-transcriptional regulation which happens in the cytoplasm. And it is more like how the mRNA is degraded or the protein is degraded or the protein. So you have these different levels of regulation. Also, the RNA epigenetics is also there because they are the methyl groups, so the acetyl groups can also bind to the RNA and regulate cis gene expression. I just gave you a brief snapshot because we will be discussing a lot about this. It's just to revise ourselves where and give like a picture of the cell, the different levels of gene expression which might happen in a cell. And now, we will come to that later, but this is one technique which I would like to discuss, which I first optimized during my PhD. This was one of the favorite technique which I later applied in my postdoc as well, and I think it will be nice to discuss this. So this is more about proteins. So it's about when you are studying, it's called proximity ligation assay. In short, we call it PLA. So this is based on if you want to study the protein-protein interaction in your cell. So say if you have protein A and if you have protein B. So the most common approaches we usually take is immunoprecipitation. So you have a protein A, you want to study protein B. So you immunoprecipitate protein A and then do a Western blot and you see that whether protein B is coming up. That's a very standard age-old procedure. But these are the upcoming procedure. So the advantages of this is that you don't need a lot of cells because for immunoprecipitation, you have to culture a lot of cells or bacteria or your culture. You need a lot of protein to do that. It's time consuming. It doesn't give you the visualization. So here in this case, you can visualize under the
So
microscope.
it's co-immunoprecipitation or immunoprecipitation for protein.
It's a co-immunoprecipitation, yes. So in this case, you can visualize this under the microscope. You don't need a lot of materials. So if you have a cell or if you have a bacterial culture, you know which protein. So first of all, you need two antibodies. So if you're targeting protein A and protein B, so what happens is that this kit is readily available in Sigma. It's called Duolink proximity ligation assay. So what you do is you have two antibodies which you have to optimize, which can stain your proteins. And after that, they have the probes, plus probe and a minus probe, which will bind to your antibody. So when the proteins if they are close and if they're interacting, what happens is they give you in the kit, you add some DNA ligase, you add some enzyme. What happens is that then this probe circularizes. It's called rolling circle amplification. And then they give you the primers and they form like signal with red fluorescent dots. And when you observe under the microscope, you can see that if these two proteins are interacting, you see these red dots. And if they're not interacting, you don't see anything. I'll just show you some of my results. It's not very clear. This was back in my paper in Oncogene from 2017. So I was, these are all colon cancer cells. So HCD, SW480, HT29. So I was studying two proteins, P68, the RNA helicase, and I was trying to see if it is interacting with STAT3 because I was trying to prove that that's a co-activator of STAT3 in regulating its gene expression. And there where you see that in the nucleus, this is all stained, blue is all the nucleus stained by DAPI. You see there are red dots. And then you can do the calculations and you can do the graph. You can quantify the red dots by Imaris or imaging softwares. And it's really a very good quantitative technique which you can do. And so yeah, that's about it. And I think that is a good technique which I learned. Coming to another technique which I did in my PhD was DNA affinity precipitation assay. Which I did a lot of modified forms of that assay as well, which I will discuss shortly.
Momita,
So-
you mean this co-immunoprecipitation is better than 2 hybrid or that bisynchronic BIFC assay also?
Yes, sir. I think co-immunoprecision is better because in the East to hybrid system, it can give you good resolution. But I think if you are wanting to do those kind of, it's better to do this dual link assay because it is more sensitive. Even if your protein is very low and the interaction is very low, you can still quantify the dots.
So this
So
paper,
antibody should enter inside the cell, then only you
Yes.
can.
Yes, the antibody should enter inside the cell.
So it is very heavier because even single antibody cannot enter easily. We have to, Hana.
Yeah, you have to really optimize that.
It's difficult.
कैसे डालू रे डब्बा नहीं लेके जाती तू
this
Komal Maurya,
initially-
please mute. Why? Komal Maurya.
Momita, this is published initially in which journal? This technique?
This book was published in 2017 in Oncogene.
Oh, wonderful. Okay, please go ahead.
Yes, thank you. And then this is another technique which is really good. So this is called DNA affinity precipitation assay. So this you use when you think that you have some, you're regulating, say in your culture, microbial system, or if you're working with eukaryotic system, if you think that some of the proteins are binding to your DNA, okay, and you want to know that if protein A is binding to a region of your DNA. So how you do it? One, we do by very standard procedure called chromatin immunoprecipitation, which I will not discuss because everyone knows that. This is a much more refined technique and much more simplistic. So say you know already the regions of your DNA.
Not only
Say if you want
chromatin
to-
immunoprecipitation, DNase-1 footprinting is also used to determine where your protein is binding on the DNA.
Yes, exactly.
That is in vitro experiment.
In vitro experiment.
Yes.
So this is one of the thing where you know that where your particular protein is binding. So basically you design probes. It's very easy. A simple probes you design across your DNA. So you know that this is my region where the protein might bind. It can be anywhere. It can be in your. So then you tag it. You can ask the company to tag it with biotin. So you incubate your either your microbial extract or your cellular extract with the biotin label probes. So all your probes, so all the DNA which you have incubated, now whichever region is binding has the biotin probe. Then you add streptavidin beads so that then the streptavidin bead will capture the biotin probes and you pull it down together. And then when you do a Western blot and try to do your specific protein, you then see in Western blot that your particular protein is bind to that region of your DNA. You can mutate the sequence to see that, okay, this sequence is important for this protein. If I mutate this sequence, the protein doesn't bind. It's very fine technique. It's very non-hazardous as well. And it doesn't need any radioactive and it's quite straightforward. I think this is one technique which was really good. This is also published in
So it
Oncogene.
is also old technique means IMSA we generally use. If our purpose is not to determine the sequence, then simple technique is IMSA, electrophoretic mobility shift assay.
Yes, just to see the shift. Yes, absolutely.
Yes. And that is easier. No need of Western blot.
Yeah, that is even easier. You just run. But here, if you want to know which protein is binding, you can just do the Western blot and see that that particular protein is binding. Or you can also send for sequencing, and then they can detect which amount of proteins are bound in your DNA.
Yes, please.
Coming to the next slide. So this is one of the results which you can see that I was working that time with the region I was trying to see whether P68 and STAT3 is bind to the STAT3 gene promoter DNA. And I have used the wild type probe and I've used the mutant probe where I deleted the STAT3 site. And you can see that P68 is binding stronger. And when I mutated, the binding was much lower in the site where I deleted the STAT3 part, which showed that how important the STAT3 part was, but to bind P68 in that region. This was also published in Oncogene.
But the STAT3 is also absent. STAT3, it should come, na?
Yes, STAT3, because if you delete that part, so in the wild type, you can see STAT3 is binding, but when you delete the thing, the STAT3 was not there because I have deleted the binding site.
Okay. Then why that amount is less in upper?
Yeah, so when I did a lot of work, there was another factor as well, which
Sometimes if we reduce the amount of one, है ना, beta or prey, then other
yeah.
thing will also reduce accordingly.
Yes, so because P68 was not only binding through STAT3, there was another factor as well. So if you do those, I didn't show them details. If you do the double mutation, then it will go like blank.
Okay. Yes.
So, okay. Now, coming to non-coding RNAs, which we were discussing, because we will be talking a bit about those techniques. So I already told you that we have this DNA replication, the epigenetics, and I already discussed the non-coding RNA, that is mainly the microRNAs. You have nowadays the circular RNAs, which might influence, and as you see it, LNC RNAs are the long non-coding RNAs, which are very, very famous. A lot of people are now working in COVID-19 and other microbiology and also in mammalian biology as well. And microRNA, of course, the age old. Now I've been working with circular RNA as well. These are all for regulating the RNA transcription. For your microbial systems, as we know that there are no like standard microRNAs, but you guys work on the one which is called the regulatory RNAs, which perform similar function in the the prokaryotic cells as well. So it can change the translation, mRNA decay, transcriptional regulation, mRNA regulation, so these all techniques can be applied across prokaryotic and eukaryotic cell. Now I will be talking a bit more about sequencing work. So this is a very age-old technique, we all know, but it's important to discuss because a lot of the future techniques I will show is dependent on this one. So we all know that, the human genome has been sequenced. A lot of work we do now is based on RNA sequencing. So what is basically RNA sequencing? The crude language, the very base level RNA sequencing is called bulk RNA sequencing or total RNA sequencing, where you can have DNA from anywhere, any species. So initially what you do is you isolate the RNA and then you do a cDNA synthesis because RNA is not stable. Then you ligate the cDNA with different adapters and barcodes. You do the library preparation. And then according to the barcodes, you send it for sequencing and you bioinformatically analyze that. And then what it gives you is the difference of transcriptome, which we call transcriptomics. It gives you overall changes in all of the RNA in your cell. So you treated cell A with nothing and you treated strain B with, say, some treatment. And then you want to compare what is the overall change of your whole RNA or the transcriptome of the cell, and then you perform the RNA sequencing. Now, this is very standard. Companies, even in India and all over the world, can perform it in a very, very low cost. And it forms
RNA sequencing is different than DNA sequencing, or only the one step which convert mRNA to cDNA, or any other?
Sir, it is bit different because for DNA sequencing, you get the changes in the DNA. The workflow is the same, but for RNA, you specifically then fish out for different kinds of RNA and you get rid of the genomic DNA because you perform the genomic DNA removal and the DNA removal.
Means
And you also,
there is no way to sequence directly RNA. We only sequence DNA ultimately. Am I right?
that is true. No. Now there are companies. I have a friend who has started his own company. In America, he's spreading his branches in Europe and in India as well. His name is Prabhu Mathiyarajan. He's from South India. So he has done a phenomenal work of direct RNA sequencing. So
Okay.
there are technologies now where you can perform direct RNA sequencing.
But
It's
mostly people sequence DNA, है ना?
Yes, mostly people do sequence DNA.
Yes, means
But
they
now
convert
there are
mRNA to DNA and then they
yes,
convert.
DNA is more stable.
Yes.
Yes. So I will just show you some of the techniques which I have done in my last paper. So what we did was as you can see, we collaborated with clinicians. So we have control hearts. We have hearts with diabetes and ischemic heart disease. We collected the the biopsies from the heart, very tiny tissue. And then I performed the RNA isolation, everything we sent it for sequencing. And then we collaborated with the bioinformaticians. So we generated a network. Our idea is to identify new forms of novel RNA species, which are different between the normal heart patients and the patients with diabetes and ischemic heart disease. So in the whole thing of that's what is one of the benefit of RNA sequencing. We started the whole project with RNA sequencing at the first. And then we found out a top candidate, which was changed, was some circular RNAs. And I'll just show you this. So we eventually I discovered a circular RNA. It's called NPHP1. Now, it is important in kidneys, the linear form. I will discuss what is the circular form. The circular form is a bit different. So it's just to see one example as I show you here. This is one of the angiogenesis assay of the heart. So it's like blood vessel formation. So you see when you knock down this circular RNA in the heart, the blood vessel formation is highly reduced, showing that how important it is for the generation and cardiac regeneration. So we found the circular RNA, but bioinformatics can only predict, but you have to validate in the lab. And there where your other techniques are important. So what are circular RNAs? I'll just briefly tell you. Circular RNAs are very novel classes of RNAs. It is very new field. It can regulate gene expression in a high level. It can degrade proteins. It can regulate mRNAs. One of its function is to bind to microRNAs. So what it does is, as I discussed, microRNAs can bind to your mRNA and silence the mRNA. On the other hand, circular RNAs can bind the microRNAs. So if circular RNAs goes and binds all the microRNAs, your microRNAs cannot silence your mRNA. So that's the whole path. It's called process called sponging. So it sponges and binds all the microRNAs so that your mRNA is free to translate. So this is another very interesting protocol. The protocol is initially actually optimized in India. It's one of the labs in India, but it's a very nice protocol which you also recently published in our paper. It is circular RNA pull down. This is RNA pull down now. How you do RNA pull downs. So in circular RNA pull down, which is similar to DNA affinity precipitation assay, because circular RNA is circular, it is not linear. So it forms like this. So you design a probe in the junction, where it folds. And then because that's the unique region of the circular RNA. And then you tag it with biotin. Eventually you use streptavidin beads. Again, it will bind all the circular RNAs which you have. Then you also have the your specific circular RNA, the probe you used. Then it will bind all the microRNAs which are bound in your circular RNA. And you isolate the RNA, do a standard PCR, generating primers for your microRNAs, and you can then detect whether that particular microRNA is bound to your circular RNA. Similar way, you can use this technique from different RNA immunoprecipitation species. So if you are working with circular RNA, a pi RNA, or a long non-coding RNA, and you want to see whether it is interacting with another species of RNA, you can do this kind of technique, design a probe, attach with biotin, and then try and do a PCR for that other RNA species to see that whether it is really interacting with that RNA species. This is one of the results which we have published. It's just a few months back, we published this paper in Journal of American Cardiology, Basic to Translational Research, and where you see that here we have used two different probes, and you see that the circular RNA is successfully bound, and we also have one of the probe didn't work for the microRNA because it was too big, so it needs a lot of optimization. The other probe, which was more optimal and smaller, was very successful in pulling down the microRNA together. We did a lot of we screen all the other microRNAs as well, and it was not positive. So this is one of
the circular RNA you find only in eukaryotes or in prokaryotes also it is there?
In prokaryotes as well. It's a very new emerging field. So not much information is available, but you can find it in prokaryotes as well.
Oh, wonderful. And when it was discovered, by the way? When this circular RNA was discovered?
I think not too long. I think maybe 5-6 years. Then there has been a lot of work, but mainly focused on cancers and stuffs.
Yeah,
So
okay.
for the cardiovascular system, it was quite new.
And now commercial available kits are there. That's what you mean?
Yes, commercial available kits are there. You can standardize the work. It's quite nice.
Wonderful. Please go ahead.
And then, so eventually we do the reverse pull down as well. So we used the microRNA similarly, as I discussed, to tag it with biotin. So basically, when you are studying interaction between two RNAs, you can design probes specific sequence, you can tag it with biotin. It's very straightforward, one-day work, and you pull it down, but it requires clever planning and designing and clean experiment. Then the same way you pull down, and you see here I have pulled down the microRNA 221, and we have successfully saw that the circular RNA is binding to it. So, and eventually we proved, then we worked together, what are the downstream genes of this microRNA. So you can see it's VGF, and VGF is a very important factor for angiogenesis, that is in the heart for blood vessel formation. So total together, the summary of this work was that the circular RNA was helping via this microRNA to produce more and more blood vessels in the heart, and is important for cardiac regeneration. Okay. So another part of the work which we were doing is methylated RNA sequencing. So as I discussed that now you have you can have methylated groups or acetyl groups which can bind to your DNA or your RNA. This is more about the RNA work. So and then it can change the gene expression, because if it binds, it can blocks or it can recruit other factors which might degrade your mRNA. And this way you can perform the sequencing. This kit is also commercially available in Sigma. And this process is called methylated RNA immunoprecipitation. It took us a while to optimize this whole technique. It is called MeRIP. So it's RIP is for RNA immunoprecipitation and Me is for methylation. So basically you have the total RNA, then you isolate the mRNA, then you fragment it. You have to fragment it. And then you add this commercially available antibody against the methylated protein. You immunoprecipitate it, and then you prepare the library, and eventually you send it for sequencing. And these are the results and you have to also parallelly do the normal RNA sequencing to compare the enrichment, the methylation enrichment and the general thing. As you can see peaks like this. This is the results of my colleague, the bioinformatician. You can see these are the two genes where you can see that these were highly methylated, and you can see the peaks. Say one example is LET7A1, and you can see by RNA sequencing that the expression was really, really low because it was methylated. So this is also one of the very good technique which if you are studying the epigenetics of your DNA and RNA, you can use this kind of technique in your system. Now, these are the interesting stuffs. Okay, now shifting the picture from more like zoomed in techniques to more bigger techniques. So this is the talk of the town, single cell sequencing, which I have done a few years ago. It's the most emerging technology now. So what is the difference between RNA sequencing and a single cell RNA sequencing? So single cell RNA sequencing, individual cells are physically separated. So when you do the RNA sequencing, it gives you the gene expression changes, but it doesn't tell you which cell. So say if you take a liver tissue, or if you take a E. coli strain, so it gives you the changes of the whole culture or of the whole tissue, but it doesn't tell you which particular fractions of cells are affected. So here by single cell RNA sequencing,
people are working on single cell omics.
Yes, exactly. So here, you can have a higher resolution. So your individual cells, say I was working with heart and the brain. So our work was to use the heart and the brain of diabetic mouse and normal mouse. So healthy mouse versus diabetic mouse, and to see how the gene expression is changed in the single cell level. Now, if I do a normal RNA sequencing, of course, there will be changes between the normal and the diabetic condition, but it will not tell me which particular changes are coming from which particular types of cells. In the single cell technology, so in the heart, you have different types of cells. You have cardiomyocytes, you have endothelial cells, you have other types of like fibroblasts. So when you do the single cell sequencing, your individual types of cells are segregated. And then when you finally analyze the data, you get the gene expression change from individual cell types. And similar for brain, you have brain endothelial cells, you have parasites, you have oligodendrons, several types of brain cells. Similarly for any cell system. So what it does is that it's very highly optimized by 10x platform and other companies in India as well. So basically you load your culture and then you have a gel bead. So your gel bead coats your cells, and when it enters the machine, it separates individual cells. and it separates individual cells. As you can see here, its individual cells are separate. Then your library is generated, and when you analyze the data, you see your data, the gene expression is from individual types of cells from different colors. Yeah. So like say this is one of the thing which we're working from the heart, and you see this is the UMAP plots from healthy human hearts. You see you have different population of cells, adipocytes, cardiomyocytes, endothelial cells, fibroblast cells, and this single cell resolution gives you the gene expression change across all the different cell types, which you can specifically point out what is happening where, not exactly like whole expression. So it gives you the single cell, and this is a very highly sought after technique, which we also perform the single nuclei. So single cell and single nuclei is the same thing, but for the single cell, your cells has to be alive. For the single nuclei, you can freeze your samples for long term and then do your sequencing. So it helps the time optimization and all, and it gives you similar results unless you want to really see your cells. But if you just want to see the changes in the gene expression, single nuclei is a much better option because you can freeze your samples. And the latest, the most hot topic which is going on now is spatial RNA sequencing. This has come to the picture for the last 1-2 years. It's called spatial transcriptomics, or the spatial RNA sequencing. So this is where you have your tissue, or you have your, so you say if you want to study a tissue where your microbes are being affected, say if you're working with Helicobacter pylori, and if you want to see how your liver is affected, so, and you want to study the gene expression, in that case, it will give you the whole. So basically you do the microscopy. You have your cells, they have different like arrays and barcodes. It's like a microscopy based, it's a joint technique combined with microscopy and transcriptomics. So it will give you a high resolution. So you will be able to see different tissues in your organ, and the whole single point gene expression changes in all of your organs. This is the most talk of the town now, and a lot of people are trying to understand how they can perform. It's already optimized, but they're trying to implement that in their own group, spatial transcriptomics. You can see this is the cover page of the cell. And this is how your result looks like, So this is a particular tissue, and you can see all the different colors that the gene expression changes from each individual cell types. The only disadvantage is that you have to still fix the cells, because this technique is still under optimization by very, very big labs all over the world. But it gives you a lot of good information, and a lot of people are really, really enthusiastic in working on this kind of techniques. So the future of gene expression, molecular biology, is multi-omics. As we know, we started from RNA sequencing, single cell sequencing. We are coming to literally spatial transcriptomics, and the more future is coming is all multi-omics. But always, all this gives you a very good high resolution at a go, but still it's very, very important to validate everything, to understand and find that one molecule which might really help. So the scientific part is done, and now we can, you can apply all of this in different field of your work. And let me know if you have any doubts, if you need any knowledge, if you need any papers, any protocols. I will be happy to share and help. Regarding careers, you can move this for the students and the researchers. If you, of course, a standard way to move out to pursue higher academic training is whether you want to do a postdoc in India or outside. Both are equally good. Government of India has good schemes now. If you want to stay back, if you want to gain international exposure, of course, you can go to UK, US. UK has good opportunities as well. The work-life balance is good. And there are amazing universities from Oxford, Cambridge, to a lot of good universities in London, like Imperial College, King's College, University College London, Queen Mary University, University of Sheffield outside London. You have good institutes in Scotland as well, like University of Edinburgh, University of Manchester as well nearby, University of Glasgow, and then you have a lot of good work culture. a very good thing in the UK is you have a lot of networking, so you get to know a lot of people, and then after that you can apply for fellowships, grants. You can return back to India. You can stay back outside, follow a traditional academic route. Applying for fellowships for grants, these are always, of course, always very competitive. You can pursue that. Other than that, in terms of UK, you have very good biotech firms, lot of startups here. A lot of people have started their own startups. Especially like they have a very interesting startup here. It's where they're trying to replace dairy, because a lot of people have all this intolerance to dairy with so they're trying to produce recombinant DNA in E. coli, how they can produce milk from E. coli. Something a recent, a very good startup has been hugely funded here. They've already started
Zero
work.
Cow Factory in Surat. 4
Yes,
million
yes.
fund. Yes, Zero Cow Factory. That is
Yes,
$4 million startup company. They're trying
that
to
is
produce
great.
recombinant casein protein.
Exactly. So this
Yes.
is really going a lot of good work. So yeah, there's a lot of good startups, biotech firms. You can have your own startup. After you gain some knowledge, you can have a startup in India. You can start up in India, startup outside. You can still be based outside and run joint companies in India and outside as well. So that's a good option as well. Other than that, you can work in charities and governments, directly community engagement. You can work at as a civil servant. You have very good officer roles in public, public service sectors, where you have to, like, do like surveillance reports, analyze the public health concerns, and work for the government to help the government to understand the public problems, the health and safety. So there are a lot of career opportunities apart from the traditional academy. Like, you can work in companies, you can be an entrepreneur, you can work in industry, you can work in government offices for as a scientist for different public health concerns, which are also equal. Also charities like Cancer Research UK, breast cancer, prostate cancer, dementia, Alzheimer's. Apart from that, you have all this infectious disease based charities as well, general well-being communities charities as well, which you can work, which is quite nice. So I think there is lot of so one thing is always keep yourself flexible. Nothing is like the best, everything is best. You have to do your best, and you can get everything from your from you can learn different things. So as I say that the key to success is, of course, strategy, focus, goals, growth, innovation, and focus. And never lose focus. Always keep focus. Your focus will change. Your dreams will change, but never forget, how much. Always make your country proud. Be confident, and always never let anyone put you down. That's very important, because and always move forward with a dream. Keep your mind positive. Live with integrity, and make other people feel important. Also empower others and build your self confidence and go ahead, charge yourself. With this, I would really like to thank all of you, the Microbiology Society of India, for giving me the opportunity. Of course, my PhD lab, my PhD supervisor, Dr. Minal Ghosh, Malini ma'am, and of course, all of my lab mates with which I started my PhD journey. I learned a lot from them. After that, I moved with Professor Neil Dillon. I learned a lot. He was my initial mentor in the UK. He was very strict, but I learned a lot from him. My lab later on, my group, Costanza Emanueli, and all my colleagues here, especially Dr. Prashant Srivastav, who is now Associate Professor in Imperial College London. He's initially from Delhi. He did his PhD in Germany, did his long postdoc in brain sciences in Imperial College, and now he has been a successful professor in Imperial College as well. He's a very good bioinformatician, and he has been my mentor and my brother, who has helped me like a lot. And other people has been amazing. I learned a lot from him. And yes, and not better the least, I always believe that at the end of the day, we have a global soul. I'm a global Indian. My heart always belongs to India. My soul always belongs to India. Wherever I live, I will always try to make my country proud and always try to give back to my country. And Bengali's picture is always incomplete without Ma Adi Shakti. So with her blessings, I would, of course, continue my journey forward. Thank you, everyone. And I wish you everyone a very, very, very happy evening.
बहुत अच्छा.
Very good presentation.
Thank
Yes.
you.
Yes, बहुत अच्छा.
Inspiring.
So what is super postdoc, madam? Super postdoc terminology is dominating nowadays, especially in case of USA.
Yeah, well, super postdoc is like, people often, which I don't believe, because it's like,
Go on
it's
doing one other postdoc.
controversial. Sometimes people are making it a toxic culture, like saying like, you go for their few years, you publish amazing papers and you get like a, but I think, I've seen it's not dependent on your luck as well, because I can give you some examples. There was one person who used to work with me in IICB. He used to work, he's a microbiologist as well. He's my senior. And he got into Snyder's lab in MIT, straight from IICB. He was very happy. He was like, okay, I'm going to Snyder's lab. So after he went there, the lab was of 50 people. He was lost. Like 2 years, I'm completely lost. I couldn't do any science. But what he did was he did a very good networking. He got into a Pfizer. Now he's in a very good position in Pfizer. So I think it's just using the situation, because and being happy, not to we can do science
So
in lot
what
of
do you
different
suggest after completing PhD from India, people should opt postdoc outside India, or they should search job here in India?
Sir, I think it's a very good question. I think because at the beginning, before 10 years, everybody is like a norm. Let's go. I think now government of India is also giving lot of good opportunities, good money as well. I think it's very important to understand what they really want and what are their future goals. So, say if they really want to, if there are family reasons, or if they want to stay back, it's still a good opportunity. They can write for independent fellowships, or they can go for a short postdoc, say 2 years postdoc in US or UK, or even in Europe. Say Germany is very good. Germany is very good for short postdocs, and even Switzerland, they pay a lot of money, and then you can go back to India. Then,
Sir, many student fresh PhD ask this question: ki whether we should go in industry in India or search job in India, even in private university, or we should go for abroad for postdoc. That is very tricky answer. We cannot give a straightforward answer.
yeah,
That's
it's a
very
very tricky answer.
difficult.
If they want to continue to industry, I would say that they should stop doing a very long postdoc, because that doesn't help. Because very long postdoc, industry doesn't count that experience. So when you apply, they say that doesn't matter. Even if you have worked 7 years, 8 years in an academic environment, we don't consider your experience, because don't have industry experience. So it depends what they want to do. If they want to join industry, either they can start applying to industry right away in India, or they can do a short postdoc, 2-3 years, gain some good experience, and then come back to India, or stay outside and apply to other companies. If they want to only become a PI, they can go for a long postdoc and apply for, or if they want to have other options. Otherwise, doing a very long postdoc can be a bit dangerous for
What
them
is
to.
the observance as a faculty in USA after postdoc? What is the you can say probability or percentage people get absorbed there as
Um,
a tenure track faculty or full faculty?
Yes, sir. In USA, the absorption is still quite good, though there is a political unrest. And in UK, comparatively, it is good, but UK is more competitive. But because you have a lot of European people, well as here, in US, you still have better tenure track grants. In UK, it is still possible if you have very good papers. But now a lot of people are moving out from US because of the political situation. But usually the opportunities are quite good, because recently there was a position for 20 people as assistant professor in Imperial College, and in among the 20 people in my department itself, there was 8 position, and in the 8 position, 5 people were from US who already have labs in the US. So they are trying to move out from the US and go to other countries because of the political, and most of them are Europeans. And for Indians, Singapore is also a very good option. The absorption, if you publish good papers, Recently, another Indian made me very proud. He's from Lucknow, Dr. Anand Sharma. He did his very short postdoc, like 4 years of postdoc in Switzerland. He got an assistant professor position in Imperial, but then in Imperial it was okay. He was permanent. He was happy, but he got a very super offer with I think 5 million pounds from Singapore to start his own lab. So he left Imperial and now he has started his own group. He's just moving in this month to Singapore for.
Please share your LinkedIn page also in chat box so that we will connect you, है ना?
Sure. I will just now. I
Chat
will.
में लिख दीजिए तो, है ना? Because my PhD guide was also Bengali and my senior, आप जो कहानी बता रहे थे कि सीनियर गाली देते वो करते, तो हमारे सीनियर भी जो थे ना PhD में, अरे तुम कुछ नहीं आता, तुमको कुछ नहीं आता. He was MBBS and then he joined as a PhD scholar after MBBS कोलकाता
Oh
से
dear.
ही, है ना? और उन्होंने भी हार्वर्ड में किया. स्टिल ही इज वर्किंग इन सम, है ना, प्राइवेट इंस्टीट्यूट इन छत्तीसगढ़, जगन्नाथ पाल.
I see.
Dr. Gopalji,
जी सर, जी सर.
you have asked about the opportunities outside the India
यस सर.
percentage. So as per my knowledge, because I know now our Indian student in every country.
यस सर.
So in USA, still there are better opportunities. Only waiting period now is more.
Yeah.
यस.
That student should have a patience. And as she has told, political situation doesn't allow to stay in USA without job.
Yeah.
Yes. Yes.
That
sir.
is a now point why people are leaving USA.
Yes, sir.
And as she has told, in Singapore, even in Bangkok, you see, in Asian Institute of Technology, I found about 50% Indians are there in Asian Institute of Technology.
सारे
Even
टॉप इंस्टीट्यूट इंडिया में PhD करके सब पोस्ट डॉक ही जाते हैं सर.
even in China and in the Japan, many Indians are there. So there are opportunities. Momita,
Yes, sir.
really I am proud of you
Thank you, sir.
because you have explained your journey very nicely and you have touched to the subject also
Thank you,
very
sir.
excellent manner. But
Very
the only my frustration is that only about 30 participants have joined. Actually, I have
मैंने ग्रुप में भी डाला तो 4-5 बाद में और आया, देखा मैंने, सोचा इसके बाद सीखना ही नहीं चाहते.
लेकिन मैंने लिखा है अभी, all other are busy in asking the question. They will ask question tomorrow to me, sir, is there opportunity outside the India? वो परेशान करते हैं सवाल पूछते पूछते, लेकिन they will not attend such courses.
Yes. जब लोग आ रहे हैं expert, then they don't attend and then discuss.
We have to actually tell them that they should attend the lectures when NRI faculties are
Correct.
So that
How
is,
nicely Mamta has explained.
Yes, very good.
she told her
Yeah.
PhD experience, then she wanted to come to Bangalore for MSc. Then she told that microbiology was not there in Kolkata or West Bengal. Then the
Correct.
IICB was the actual lift for us to come in the research. So that was really
Yes.
that's actually even I wanted to know. Of course, she did not to answer perhaps. How is the working environment in CSR lab, especially in IICB, and where you are working? In a very
Yes.
briefly, you can tell.
To be honest, sir, I really loved working in IICB. I think हमारे इंडिया में जो है ना, we have one thing that is collaborative working. And
Exactly.
there is competition, but we used to help each other a lot, like inter labs. Like, my whole cells, you see, all the colon cancer cells, which I worked in PhD, none of them we bought in our lab. It was given by neighboring labs. Yeah. And nobody will say, okay, give my name in the paper. I learned a lot of things from other labs as well. And it was very, very good in the here in Imperial in London. It is very good as well. But I think there is this warmth in India, which you still miss. Like, there is this informal help. Chalo milke karte hai, that's the thing. In UK, there are different. In UK, the rules and regulations are very good. There is still the global life. Like, you get to meet a lot of new people from all over different parts of the world. You have British, you have Europeans, you have Africans, you have a lot of Middle Eastern people. Like, I have a friend who is one of the very elite scientists from Qatar. She's also, so you get to know people from different parts, so that is also very advantageous. In UK, we also talk a lot about well-being, to cut out toxic culture, bullying, and all this of things. So UK, in this part of the thing, I learned a lot from UK, to fight for oneself, because Indians can be targeted as well, because we are good. So, I have seen a lot of like semi-racism and this kind of things can target, because we are good. People do get jealous. And then how to defend oneself.
That kind of problem one can face even in Germany also.
Yeah. So
Even
those
with
kind of
the-
things.
The experience in Germany is really not that enjoyable or that easygoing. But
सर
then
रेशियल थोड़ा रेशियल डिस्क्रिमिनेशन थोड़ा था यूएसए में सबसे कम है
Yeah.
यूरोप में ज्यादा है यूएसए में सबसे कम है क्योंकि पूरा यूएसए तो बाहरी लोगों से ही भरा हुआ चाहे इजराइल हो दूसरे हो
But if we
यूरोप में
manage
ज्यादा
1
है
or 2 years in place like Germany, then you can manage anywhere in the world. That one
True,
good.
true. Yeah, I have heard that. Especially against, against minor communities, I have heard like Germany is really, really bad. To other countries as well. Indians are still okay, but like I think they target Pakistani people very, very bad.
कुछ लोग जाके अपना नाम भी तो कर देते हैं अभी तो यूके में भी तो थोड़ा सा नाम खराब कर रहे हैं पाकिस्तान
हाँ, that has.
Dr.
Leave,
Mohammed, Dr.
you
Moses
see,
are there,
Dr. Gopal ji,
जी
Dr.
सर
Gopal ji,
जी सर
we will not discuss on that issue.
yes
Come to
yes
the
yes sir.
academic. Yeah?
That's why I'm not going sir
Correct,
Sorry.
correct. So, again, I will ask Wasim sir or Chandatri ma'am. Now you conclude it, and this lecture will be circulated. And I got message from few students, sir, please circulate this lecture. So I will circulate it.
yes इसी का लिंक दे दीजिएगा watch it
Correct.
yes sir तब नहीं वॉच
Okay,
करेंगे then they will not return back है ना सर ऐसा है बच्चे का है ना वॉच नहीं पूरा
Chandra,
करेंगे
Oh, yes, sir. Wasim sir going to conclude the session.
Okay, thank you, sir. We are thankful to Dr. Deshmukh sir for attending this lecture. We are also thankful to other dignitaries who listen carefully and make the session very interactive. We also thankful to all the participants for listening this lecture. As well, we also thankful our chairperson of this NRI lecture series, Dr. Chandratri ma'am. And lastly, we also thankful our resource person Dr. Momita Sarkar ma'am for sharing his insights in advanced molecular techniques. So, with the kind permission of the President, Microbiology Society India, we declare that lecture is over. Thank you very much.
Thank you.
Thank you all.
Thank you, sir.
Thank
Thank
you.
you.
thank you
Thank
so
you.
much
Many eminent teachers today attended, including from Aligarh Muslim University also. Many have attended. Thank you.
I inspire everyone from India, and I wish all the best to all the students. We all make ourselves proud. And, everyone from all corners of the India, from everywhere, wish you all the best. And all the best to this.
Very good. Very good.
Okay, thank you all.
Okay.
Thank you.
Thank you. Thank you so much.